Hello everyone and welcome back to Willow's Notes. In today's video, we will cover everything we need to know about meiosis. We will use this 2n = 6 sample where we have 3 paternal chromosomes and 3 maternal chromosomes. So 2 sets of 3, which is what we mean when we say 2n = 6. Just like in mitosis, by the time the cell is ready to start meiosis, DNA replication has already taken place. And now each chromosome is made of 2 sister chromatids. So let's start with the first phase, prophase 1. Just like mitosis, the nuclear envelope breaks down, the chromosomes condense, the centrosomes move towards different ends of the cell and form the spindle. However, something very different from mitosis in prophase 1 is that the homologous chromosomes pair up. They align gene by gene next to each other, forming what we call a tetrad. A very important event takes place in prophase 1, which is right after the homologous chromosomes align. The DNA of the 2 non-sister chromatids, one maternal and one paternal, breaks. And the non-sister chromatids join to close up the breaks, causing crossing over to take place. So now the points where the crossing over took place become visible as chiasmata. The homologous chromosomes, they remain attached to each other, but they move slightly apart, which makes the chiasmata more visible. In each homologous pair, there should be at least one crossing over so that they can stay together. The crossing over sticks those homologous chromosomes together because remember, sister chromatids are already connected through their centromeres. The next phase of meiosis, which is metaphase 1, again, just like in mitosis, the chromosomes align at the center, what we call the metaphase plate or the equatorial plate. The only difference here is that in mitosis, the chromosomes are not paired at the center, whereas here, because already in prophase 1, the homologous chromosomes paired up, now we see them paired up at the center of the cell. And each of the sister chromatids are attached to kinetochore microtubules from one side. We know that anaphase is the phase where the chromosomes separate. They move towards different poles, right? In anaphase 1 of meiosis, it's the homologous chromosomes that will separate. So as you can see, the homologous chromosomes, they separate and they move toward opposite poles. The sister chromatids are still attached at the centromeres, so they migrate together. Following anaphase 1 is telophase 1 and cytokinesis. Now each new cell has a complete haploid set of duplicated chromosomes. So it's haploid, and yet the chromosomes are duplicated because each chromosome is still composed of 2 sister chromatids. That's why meiosis does not end here and meiosis 2 begins. Meiosis 2 starts with prophase 2. The same events that take place in prophase 1 take place in prophase 2. Events like what? Events like the centrosomes moving towards the ends of the cell, the spindle fibers forming, as well as the condensation of the chromosomes. The only difference, of course, is that the homologous chromosomes are not going to pair up because they are now in 2 different cells. So in metaphase 2, the chromosomes are going to align at the center of the cell, what we call the equatorial plate or the metaphase plate, followed by anaphase 2. In anaphase 2, again, anaphase is known for chromosomes separating and moving towards the ends of the cell. In this case, it's the sister chromatids that are going to separate. The sister chromatids keep moving further apart. Of course, this is happening in both cells where the sister chromatids are moving towards the different ends of the cell. once the chromosomes finish separating, we're not going to actually see them like this. They're going to loosen up and become chromatin. But just for the sake of the video, so that we can follow their movement, we're going to keep them as such. And of course, in telophase, the nuclear envelope reappears. As we mentioned, the chromosomes loosen up, they become chromatin again. And this phase is quickly followed by cytokinesis, which is the actual splitting of the cells. So by the end of meiosis, we have 4 genetically different, non-identical cells, unlike in mitosis where we have 2 identical daughter cells. Here, the 4 cells are genetically different, and each cell is a haploid cell. And in this scenario, because we started with a 2n equals 6, here each cell is n equal 3. And this was it for today's video. I hope you found it easy and useful. Don't forget to give me a thumbs up and subscribe if you haven't already, and I'll see you in the next one. Bye!